quite entertaining for everybody, so it went black for a couple of minutes, but we got there, didn't we?
we definitely got through it.
Yeah.
So we are just ticking over 1 p.m. So we will begin the seminar. So this week is webinar 2 of the PacBio Lunch and Learn Long Read Sequencing webinar series. And today we are talking about HiFi for microbiome projects. So I'd like to introduce our field application scientist, Paul Gooding, to present to you everything you need to know about HiFi for microbiome. So go ahead, screen, etc. I will be here, everybody, to moderate any questions that come through. We'll add them in at the end. This webinar will be recorded and available on YouTube after, just like the previous recording is now available on YouTube, and the questions will appear on that recording and also in the description underneath YouTube as well. So if you miss anything, it will all be available later on. So over to you, Paul.
All right, thanks, Sarah. But actually, before we start, for those that are here, I'd just like to say a huge thank you to Sarah. Sarah's actually, this is her last time hosting some little sessions like this. She's actually moving from Millennium Science onto greater opportunities. So we wish you all the very, very best of luck, Sarah, and we'll miss you heaps. So but thanks for all you've done for these sorts of things. So yes. Sort of happy and sad stuff, isn't it? But all right, so I'm going to share my screen now and go to the topics here. Oop, there we go, and hopefully we can also see that. So so yes, welcome to Lunch and Learn. I hope you've got your sandwich and a coffee or whatever, and you can settle in and listen to me go on for a little while about PacBio and long read sequencing, HiFi sequencing. So just a sort of start page. 2 weeks ago, the first session, I ran a sort of overview of PacBio sequencing, how it works. It's single molecule, very long read, very highly accurate sequencing. You make these single molecule smart bells, circular molecules, and the polymerase reads around and round and round them, and you get very long, very accurate sequencing. So that's how it works. We discussed that a couple of weeks ago. But in the following sessions over the next month or so, and including today, we're dealing with some specific applications that you can use that highly accurate single molecule long read sequencing for. And today we're talking about microbiome projects. So metagenomics is really where we're at for today. So that's probably a better start page for everyone. So we'll talk about how you might sequence bacterial and other microbes in your samples, and this might be human microbiome, which is a very hot topic around, of course, or it could also be environmental samples, so it could be soil or water samples and things like that as well. And there's various ways that you can use the sequencing technology to get information depending on the goals of your project, and we'll discuss those. So there's sequencing of 16S regions, and then there's a sort of shotgun microbial sequencing from your samples, and we'll talk our way through those a little bit. So I'll just launch these up. So the various approaches really are here, as we said. So we'll start probably with the 16S sequencing on the left there. So that's really a targeted sequencing method, because you're specifically amplifying a 16S region out of your sample for all the microbial community that's there, and you're sequencing everything, and you're looking at that sequence in detail to get an idea to the species or even strain level, as you'll see, what's in that community and how it's made up. And there's tools for analysing those data and things, which we'll mention too. Also, you can go for just a shotgun sequencing approach. So rather than targeting 16S, you can sequence the entire genome of everything's there. So just sequence total genomic DNA that's in your sample. So you extract the DNA, you break it up into sort of bite-sized PacBio bite-sized bits, which is about 10 kb, and for these sorts of applications, and basically sequence the lot. And again, there are tools to look for genes and regions of interest within those sequences, and you can get a lot of data there without making assemblies. So there's really nothing else out there that can just go from the raw sequencing effectively to get that kind of information. And then you've got the assembly, which down the bottom right there. So you can go the full distance. Then you can do that shotgun sequencing because you've got such accurate reads. You can make the assemblies of essentially all the organisms that are in your community, and make complete or near complete assemblies. And this is also very, very useful for species that aren't culturable and can't be purified very easily. So you can actually get reference genome sequences for these very, very difficult to work with species using these sorts of approaches. So, okay, so I think some of these next slides will probably just reiterate what I've said to you already, but basically this the accuracy and read length enables you to get that species level resolution out of your samples. This is very, very difficult to do, as we'll see with some of the data with short read sequencing. And so again, yeah, we can use 16S, or we can use these 10 kb fragments of the total genome that's represented in your samples from these complex populations. There's a little example here, again, using just 16S data from a mock community. This is a sort of culture that you buy that has various bacterial strains sort of spiked in at various levels, and you can look specifically at the 16S there to see what's there, and there's a given known community presence, and then see what the sequencing data gives you, and using a tool called Data2, it actually finds in these communities not only the types of bacteria that are there, but it actually finds that often there's multiple copies of the 16S, and sometimes those copies of the 16S aren't identical within each strain. And so this gives you integer level recognition of all the 16S that's in your species there. And there's a paper from NAR back in 2019 using exactly that technique. So 16S gives you that species and often to strain level information, and as I say, there's nothing else really that can get close for that. There's full protocols and everything available. We'll touch on those, though I don't want to go into them in any detail. So there's ways of amplifying your 16S. The protocols give you some sequence information for primers you can order. There's also some kits that are available for extracting the 16S and amplifying the 16S sequences. In fact, there's a number of kits from Shoreline. You can either amplify just 16S, or I think they have a something that amplifies 16S and UTS, and then maybe even into the 23S region, so you get an even longer piece of DNA amplicon that you can sequence and get even higher order data. When you're doing your shotgun sequencing, as we said, you want to get about a 10 kb insert is ideal, and let this sequencing that goes round and round the molecule to deliver really highly accurate sequencing. So any errors that do occur because of the polymerase from reading the sequence get screened out, because you've got to set effectively a big pile up of read over read over read of the same sequence, and you can screen out any of those errors, so you get very high Q scores, way over Q20, as we'll see a little bit later on. And with that data, you can go directly to that data and look for effectively functional data within that. So the presence of genes and operons and things within those clusters. the other advantage you have is there you can find information from members of that community that are present at very, very low abundance in the samples, less than 1% for sure. But also, if you want to then make assemblies from the data that you have, yes, there's tools for doing that too. You can take your data, you can make the assemblies with software like Canu, and make these full length or very near full length assemblies of basically the entire community in your sample, the entire microbial community. So briefly, as I said, there are protocols. We'll start with the 16S side of this. There's a protocol that basically gives you all the information you need to be hands-on and go and do this sequencing. If you're lucky enough to have a PacBio instrument yourself, you do the whole thing, but there are a number of providers out there that will sequence your PacBio libraries for you, the so-called SmartBell libraries. With the newer template prep kit, version 2, so the SmartBell Express template prep kit 2, the workflow now is really, really quite straightforward in your lab to make these SmartBell libraries and then send them to your PacBio sequencing service provider. There's providers on the West Coast and the East Coast of Australia. So if you have any difficulty or want to have more information, contact myself or somebody at Millennium Science, and we can help you. So the protocol itself gives you, as I said, some information on primers. These are barcoded primers, so you can multiplex samples up to 96 samples together for your sequencing. So it gives you basically all the information you need to make those primers, extract your DNA from your samples, and do the amplification, make the SmartBell templates, and get that ready for sequencing on the Sequel 2. So a little bit of information here about 16S sequencing. Traditionally, I suppose, is the word of using short reads. You have a 16S region of about 1.5, 1.6 kb, which is out of range of a lot of the shorter read next-gen technologies to read right through. Likewise, even for Sanger sequencing too, it's a little bit out of range to go through. So in the past, people looking at microbial communities with the shorter reads would pick a region. So V1, V3 was very common. V3, V4, or just V4 was also quite common, and people had their pet targets. Each lab working on these sorts of microbiome projects had their own sort of pet region that they would sequence, and that's not without issue when you're trying to compare lab data sets across different labs around the world. So not always ideal, but just looking at the availability of sequencing right through from V1 to V9 now across this 1.5, 1.6 kb fragment, the data's shown here. The red is basically the sequences that don't give the full resolution to species data, or even necessarily to genus data, whereas the white is where you've got basically two species even to strain level identification, and depending which regions you'll sequence, you have more or less success. And as you can see, by sequencing V1, V9 right through, you have a very, very, very high level of success. You basically resolve all the bacterial clades. This is from a human gut sample as an example, but it could be from environmental samples or whatever too. And just using certain regions that are listed there have their own sets of issues. They may work for certain subsets of the bacteria that are there, but they're not particularly good for other sets. So things like proteobacteria or actinobacteria, they're not particularly good for certain things depending what you're looking at. So yeah, the only real way to get confidence is to sequence right through V1, V9. And again, an example here using one of these mock communities, sequencing against what's expected And again, you'll see that the data that you get back is extremely close to the real events and the expected mock community that was there using this full-length 16S sequencing. And this data set is actually available. It's a download. If you want that information to have a play around with the sequencing data that you get back, let me know and I can provide this link for you and you can have a look for yourself. So something to note, I suppose people consider the 16S region to be present just in the bacteria, maybe in a single copy, or maybe if there's multiple copies, but they'd all be the same. But that doesn't seem to be the case now that people have looked in more detail using PacBio sequencing. It seems that often there are multiple copies of the 16S region, but they're not identical. They have differences within their sequence, and this has always been put down to sort of artifactual, errors, systematic errors in the sequencing when it was short read or Sanger sequencing. But now that we can look in so much detail with the long read length and really high accuracy from single molecules, we actually see that these regions, that they're actually real. You've got multiple copies and they are different. And using the Data 2 software here, you can actually see these number of copies of the different pieces of 16S full length that are in the genome, and how they are actually different, how many different ones you've got, and how many copies of each one of those different ones that you get. So an example here with Staphylococcus aureus, common bacterial species, it's actually thought that it was just a single copy of the 16S and that there were just artifacts going on with the sequencing, but it's been shown with PacBio sequencing now actually there's multiple copies and they are in fact different from one another. And sure enough, excuse me, across all the full 16S, nearly all isolates that are being sequenced have found that there are multiple SNPs across the entire region, this is not really so surprising, I guess, but it's the advantage it has is that if you're able to capture that information, you can sequence and get data that's beyond just the species level. You are getting down to individual strain identification levels. So whether you've got a virulent strain of the bacteria or something that's less virulent and not posing, say, a human health risk. You've also the ability to look at the gene function there and say, well, have you got antibiotic resistance or not? Those sorts of things. So it gives you a lot of power with the data that you recover. So that's probably enough. Moving on now from the 16S and get onto the full length sequencing. So the metagenomic shotgun sequencing, as it's called here. As a general overview, there are protocols and handy hint sheets and things and best practice guides available. So again, if you need any of these, and they're on the PacBio website, but contact somebody at Millennium Science, myself or one of my colleagues, and we'll get these out to you. So the best practice guide walks you through how best to prep your samples, whether the aim of your experiment is better suited to a 16S sequencing, targeted sequencing approach, or whether it's to go for the shotgun metagenomics sort of approach, and the data you can expect back from that. There are the procedure and checklist, which are the actual protocols for making your libraries and running the sequencer, and what you'll need depending which protocol that you're following, whether it's the 16S protocol, making amplicons, or these 10 kb SmartBell libraries for the shotgun. And there's also a bundle of which kits and reagents you'll need to put together depending on what you're doing. And obviously there's the workflow in the procedure and checklist, which is getting your genomic DNA for the shotgunning stuff. You're going to shear it, shearing around about a 10 kb target. So you need genomic DNA that's good quality, above 10 kb, really above 15 kb if you can get it before you start your shearing. You then make those libraries with the Express Template Kit 2, which is really quite straightforward in most labs for anyone now. The kit's right there to do it. It's just a pipetting exercise, really, and you follow the protocol, and then you've got libraries ready to go onto your Sequel 2, and then off the Sequel 2, you've got data analysis options. Really, you can do a lot of analysis using PacBio software, or you can go to other third-party tools like we've mentioned already, Data2 for 16S, or Canoe and things for making the assemblies and things with your data. So yeah, probably just reiterating that to some degree. It is important that your DNA that you're extracting for all PacBio sequencing, you want as high quality DNA as you can, because it's long read sequencing. You need long inserts, and so you need high molecular weight DNA that you're extracting. So and there are protocols around for doing that and making good quality DNA. Also, remember that particularly with the 16S, you're going to quite heavily multiplex to reduce costs, get a lot of samples onto one run. But even with the metagenomic shotgunning, you can multiplex up to about 4 barcoded samples, and we'll see a little bit of a data example of how much sequencing you expect per sample and what you can do with that data a little bit later on. The general workflow, yeah, you've got your genomic DNA, which you're going to shear down, you're going to make your smart bell, you're going to go to sequencing and do your analysis. And there's just a little reminder on there of the way that the PacBio HiFi sequencing works that we talked about a couple of weeks ago, if you were here, how you've got that single molecule and you're sequencing around and around the molecule. So you're getting multiple passes over the same piece of DNA. And that's how you get such highly accurate sequencing. So how many samples can you multiplex? Well, it rather depends on your needs. So there's a couple of sort of examples here, I guess, and by asking questions, how many can you put on? Well, what's the estimated abundance of, say, the rarest individual in your community that you want to be able to see? So if the answer to that is, I want to see something that's only present as say 1% of the total bacterial load in a sample. Okay, so you can make some educated guesses. An example here on a single smart cell on Sequel 2, you expect somewhere around 2 to 2.5 million reads that you can get. So we'll say 2.4 million here, around that for greater than Q20 quality. You want to see what 1% of that is, so 0.01. So that's obviously 24,000 HiFi reads with greater than 220. You're going to get from a sample that's 1% abundant in your sample, given that it's an average size bacterial genome. And so from that leads on to, well, what's actually the goal of what you want? You want to see this bacterial species that's at 1% abundance, but are you just trying to detect it? Or would you want to know something about its functionality of its genes, or do you actually want to assemble a full genome? And that, of course, makes a difference to how much coverage you need. So for detection, you only need about 100 reads, so very, very easy. For that gene profiling, discovery within the genome, you need maybe 5-fold coverage. So you may be talking about 3,000. Say HiFi reads, but for total genome assembly with confidence, 20-fold coverage, that would be 12,000 HiFi reads. So that's kind of the way you step through how much you can do with a smart cell and how much you're going to multiplex. So just for the sake of being able to read it, I haven't got the 4 samples per smart cell, but we can work that out as we go, just from the numbers that are on the screen. So let's say our 2.4 million reads per smart cell ATEM. So that if you only have 1 sample on the smart cell, that gives you the total 2.4 million reads for that sample. Obviously, if you have 2 samples multiplexed together on a single run, barcoded, you're going to get about 1.2 million samples per or reads per sample, because it's split between 2. Likewise, if you go down to 3, you're going to get about 800,000 reads per sample from that smart cell. Do the maths again. If you want to go to 4, it would be about 600,000, wouldn't it? So that's where you're going to be. And now, if you're looking at 1% of reads, which we were talking about earlier, for that 1% abundance, you're going to get 1 sample per smart cell, about 24,000 reads for a 1% abundance on that cell, which is way more than you saw earlier. You need about 12,000 reads, so it's about double what you need for an assembly. So if you come down to 2 samples per smart cell, you've got that 12,000 reads. It's probably enough for assembly, which are a 1% abundant species in your sample. Going to 3 per sample, you're just outside of that assembly region. Now, you might get assembly if you're lucky, but you'll certainly have plenty for profiling, because what did we say for the profiling? We only need about 3,000 reads. So you've got plenty for the profiling. You just might be a little bit pushed to get a full genome assembly. But it'll be close. And of course, if you step down to 4 samples per smart cell, you'd be down at 6,000. Again, plenty for profiling, easy. And then if you wanted to go down to something even rarer in the population, so sub 1% in your population, so 1/5 of 1%, so 0.2%. Again, with the numbers you have, you're easily detecting even off 4 samples per smart cell, and certainly with 2 and 3, you're easily getting detection, and you're in the realms even of getting functional profiling information from those kind of reads. And so that's the power really of the technology you have with PacBio reads. Some examples. Sorry, need a sip of tea. You're all having your lunch. I'm going to have mine too. Yes, an example here of 9 different human fecal swab samples, and what they look like in terms of PacBio data. So again, around that 2 to 2.5 million greater than Q20 reads, so high quality reads, greater than 99% accurate. So that's the sort of numbers that you're getting across the board from these 9. It's an obscene amount of Q20 bases, 20 billion or something. So it's a lot of data, a lot of really high quality data. Given that we were shearing for around about 10 kb, you're probably looking at anywhere between maybe 8 and 12, the majority, because of that normal distribution within the shearing. So again, we've got HiFi read lengths going on here for the movie times of up to about 10 kb. So that's good. And in fact, when you look at the data in question, you've got actually way beyond Q20 base quality. You've actually got those bases up around nearly Q40 for everything, which is what 99.99% accurate on the sequencing, which is really quite amazing. There's nothing really comes close. So what can you do with this? You've got the long reads, you've got the high accuracy, without actually going and using the computing power and time and resources to do a full assembly. What can you do directly with the data? Well, you can look for predicted genes within the sequences that you have. And again, there's an example here of 5 of the human fecal samples, and you're looking at many millions of predicted genes. Read lengths of each gene about 1 kb, and out of the reads you're looking at anywhere between maybe about 7 to 9, say around 8 predicted genes per read from those 10 kb fragments, and then when you look at the clusters on the end there too, and just to bear in mind that a lot of short read data, you just can't get enough resolution on the short reads to be able to map confidently that data back to a metagenome. So effectively, that's just not useful for this functionality, this gene finding. So unfortunately, you're kind of wasting a lot of data there because you haven't got long enough reads. And there's other advantages to doing this sort of work with high accurate long reads. So it means that you can use a lot of these tools without any modifications at all. So also with these very hard to culture, hard to work with and type organisms, again, this technology gives you the power to look at those samples that are sort of in situ, if you like, and get effectively complete or near complete genomes. And there's an example here an unculturable bacterial strain that was found in preterm neonate gut. It's one of those important species for gut health, and but it's very, very difficult to work with outside of the gut. So, but the power of the sequencing, if you take microbiome samples from the neonate gut and do the shotgun sequencing, you can get effectively full reference genomes from the organisms that you're after. Okay. So getting, I suppose, more into the sort of the bioinformatics side rather than examples of using the data, I guess, just some examples of what you get off the sequencer, and yeah, your number of bases and the length of the reads and the distribution of the reads in the tables and the little graphs there. So you can see that you're greater than 50% going into that lighter and dark blue for your base yields and the sizes you're getting there. So you're getting well over 15 kb, 50% of your samples. If you look at that that read length density plot, again, shearing for 10 kb size insert, you're going to expect most of your data to fall within that sort of, you know, 8 to 12, maybe 15 kb, and it's just indicating there the 5 to 20 kb, and you can see the density plot there is the red, the highest amount of data is sitting nicely in that sort of size range, and really at the upper end of that size range. So you've got that 8 to 12 kb region covered the best, which is ideal for what you're trying to do, and that's borne out as well by the HiFi read length distribution on the right as well. You've got that sort of distribution between 5 and 20, with the peak at the 10 kb which you were aiming for, so that's great. Just the little table on top, a couple of things to point out there. These are the sort of metadata, if you like, that you get back from the sequencer. So it gives you your total base yields, and then your unique molecular yield. Remember, you're sequencing around a molecule, so it compresses that down to the unique yield, so the really accurate reads. It tells you how long your overall polymerase read length was, and the M50 for that. It also gives you a longer subread that you managed to get out of the data, and the P0, P1, P2 is actually the loading of the smart cell. So P1 is the active producing good data cells out of the 8 million little nano holes, little ZMWs that are on the smart cell chip, and this was loaded at about 69%, which is good, and certainly what you'd hope for loading a smart cell. And P0 is the empty wells. P2 is some of the wells that are giving spurious data, so it may mean that they've got 2 molecules inside one well, or the cell's not working quite properly there. But again, that's a very small amount. So looking at a similar table here that we saw from the 16S data, but this is using the metagenomic shotgun sequencing of a mock community. Again, you can see what the sequel has done compared to the expected composition of this mock community. Again, a very, very accurate reflection of what was expected. And if you look at these top bars, the little pinstriping lines at the top, these are recognizing species that have been spiked into these mock communities at very, very, very low abundance. So you can successfully detect down to 0.018% abundance within a mock community, and still find that using a PacBio Sequel 2 run. And again, this data, like the 16S, these data sets available freely for you to have a play with if you're into that stuff. And so, yeah, do look for it. If you can't find it, contact me, and I'll send you a link for it. So moving on a little bit about assemblies now. So rather than just going from raw data and looking for functionality, if we do go to making these assemblies, you can use a tool called Canu, which is very good from PacBio data to make these assemblies. And again, some examples here from those 9 fecal samples that we showed earlier, using Canu. There's an overview here of basically your contig M50 length and the number of contigs that you've got per sample in the red dots there. So this is an idea of what you can do with Canu to put things together. And also quite nicely, there's another tool here called Patrick, which takes that cannu-derived data and bins it per sample. So it's actually telling you how many different species basically are represented within each sample. So you're looking at over 100 species in most of these fecal samples, sort of thing in the bars there. So moving sort of on towards the end now with just a couple of examples of what you can do and what has been done with these type of data for metagenomics applications. There was a shotgun metagenomic sequencing project done in collaboration with Second Genome and PacBio together. And they were looking at environmental sample, soil sample here, and the aim of what was in the soil sample and what genes, what functionality did those bacterial communities have in the soil. So that was the aim of what to look for. And so basically it was also a comparison of the PacBio power against using short read and other sequencing methods to see what results and the bang for your buck basically that you could get. So this is sort of an overview of what they did. They were sequencing with PacBio and then splitting the data out, looking for the gene predictions and functionality from the communities, and also doing assemblies where possible, and then comparing what they got with short read sequencing methods. So let's just load these up. Okay, so they had a couple of smart cells worth of sequencing, and you can see the kind of amount of sequencing that they got back in the table at the top. So your total bases and your subreads and your predicted accuracies and the amount of total HiFi bases, etc. along the top. So yeah, good quality data. You can see from the charts, the read length again peaking around that sort of 10 kb, which is what they're aiming for this. And again, the quality of the reads, extremely high quality and accuracy from the data, which is always a good start, isn't it? So how did they go with their workflow? Well, basically they had around that 10 kb average, say an 8 kb mean HiFi read length. From that, they were able to take the predicted gene coverage and dereplicate those. So basically any copies they compressed down to single gene units, if you like. And then they were able to map those back against the contigs, which they'd also use the data for to assemble those HiFi reads into contigs. So that's kind of the circular view of what they tried to do. And just a summary of what they eventually found in terms of the total number of contigs and the lengths and things there. I won't read them to you, you can read them for yourselves, but this has been recorded, so if you need to look back at the slides to get these data, you most certainly can. And I can provide some slides for you. If anybody wants to contact us, I can provide those for you too. So some good metagenomic data, and they were able to basically get a graph of the assemblies and annotate that with the various species that were present in the soil sample. It looks like a child's drawing, doesn't it? But that's what they were finding, and we can map these clusters here. So this is using a tool called Bandage. To do these sort of almost tree-like visualizations. Very powerful. It was actually showing that not only was it getting the full genomes, it was also pulling out the plasmids that were present in some of the bacterial species. So you were getting an immense amount of data and a really complete picture of what was present in terms of the functionality and getting near full assemblies for many of these bacterial species that were present in the soil. So again, a final summary, comparing it to how it was going against the short read data in terms of bang for buck too. You can see that the long read data, the HiFi data, was really very, very good compared to the short read data in terms of what you were able to achieve for the money you spent. And also an advantage is in computing time that you didn't have to do the assemblies, full assemblies to get the data from here. You could get the functionality data directly from the sequence output itself, rather than have to get assemblies, which you would need to do for the short read data in order to get that kind of functionality. And just a final example. Hopefully we're good for time. This is a gut microbiome example between healthy and diseased patients. So you've got some healthy controls, you've got some patients with diabetes, and also patients with diabetes and cardiovascular disease. So just seeing if there's any changes or obvious differences between the gut microbiome for these patients. So each of the patients had their gut microbiome sample sequenced. And again, a similar sort of work through of data, looking from the various samples here, looking at the number of HiFi reads and the mean length that you're getting across the graph. So again, good, long, high quality data, which is a good start for these types of things, as I said. Because of the data you have, you can look for a gut microbiome, but you can also screen out any sequences that map to the human reference databases. So you can see from here that the amount of host contamination, in this case human contamination, was actually quite low in most of those samples. So so obviously good swabs taken, and the extractions and everything that following the protocols was really targeting the microbial communities, which was good. So back to Canu, doing the assembly. Again, across the samples, you can see the number of contigs that were formed from each sample, and the length of the longest contig that were able to be achieved, and a sort of idea of the amount of the contigs that were over a megabase in length. So most of them had at least some contigs of over a megabase across the samples. And then you can see when you display the graphics like this, how the different samples are altered in terms of their strain diversity and abundance across the samples. So if you put in these into the blocks, you've got the healthy, 3 healthy samples on the left, and then you've got the diabetes or the diabetes and cardiovascular samples on the right. So there are changes in the way the different strains of bacteria are represented within the gut microbiome, depending on healthy and diseased patients. And you can obviously manipulate this data in a number of ways once you have it. You can go to a PCA plot, for example, and see if you can see a cluster that takes the healthy away from the diseased, and whether the diabetics only against the diabetics with cardiovascular disease can also separate out. And as you can see on this PCA plot, that you do separate these out according to the gut microbiome representation. So just finishing off really now, just an example of the various strains that were found, and whether certain strains were considered to be beneficial and others considered to be pathogenic within the samples, and how that was reflected in the gut health and the patient health. So again, these slides are available, so if you want to look in more details at these, you certainly can. But basically, you weren't able to find all the information here. It's only a running list from the amount of samples that they had, and basically the take-home message was that they needed to sample more and more to get a clearer and clearer picture of the abundance and how that affected gut health. So the positive and the negative affecting bacteria in the gut. So just put that up there as well, which is just a joy. So I suppose just finishing off, this is just a rough idea of sequencing costs, I suppose, with PacBio for these metagenomic type samples. Note that there is list price in US dollars on the right, so it will be different here in Australia. And obviously, if you're using a sequencing service provider, they may have an add-on to the price for their services. So this is purely the cost of the kits and reagents at a list price in the US. So, but it gives you an idea of where we're at. And if you need any more information and quotes and things like this, then obviously the team at Millennium Science can certainly help you. So contact us. So an idea for your de novo assembly. You're looking at off one smart cell. If you're able to look at 48 microbial isolates, let's say, you're looking at about $70 US a sample in consumables. If you're going again from full length 16S, you can multiplex up to 96. Prices coming down to about $30 US a sample in consumables and so on and so forth. If you're going for the functional profiling that we talked about, you won't need about 5 times coverage. So depending how many you're able to multiplex together, so say 3 here in the example, level of multiplexing 3, you're looking at about $450 US in consumables. And for the full shotgun assemblies, so you're getting full assembly of all your community genomic DNA there, maybe a single sample there at about $1,300 for the sample on a single smart cell. So I think that's probably it for today. It gives you an overview of what you can do with PacBio in terms of microbiome and metagenomics. I'll just take my screen off and come back to you. So I hope that was helpful to you. And as I said, 2 weeks ago, we went over the basics of how the sequencing worked. And today, this particular application for metagenomics. And in 2 weeks' time, we'll give another one which focuses really on whole genome sequencing of plant and animal genomes, so large complex genomes, and using HiFi, long read, high accuracy, single molecule reads on PacBio for making those assemblies from complex whole genomes. So with that, I'll say thank you very much. And take any questions or anything, do feel free to pop questions in the chat or do what you have to do. There might be a few things in the chat here. Are you around, Sarah? Did you monitor while I was chatting away? Are shorter HiFi reads just as accurate? Yeah, that's a good thought, isn't it? Yes, in terms of their Q scores, they are absolutely as accurate. In fact, you could argue that because you've sequenced around the molecule more and more times, depending on your movie length, that they could actually be more accurate, but you're losing some power in their mappability because of the length of the reads. So ideally, you do need longer reads than just say 5 kb in this example in the chat. So I think that's the answer I'd give you for that, Joseph. Yes, they're just as accurate, but you lose some power in terms of the read length. Longer reads are very helpful, and certainly we'll talk in 2 weeks' time about sequencing, whole genome plant and animals. You need ideally some really long reads to help get context together. So it's not just about the accuracy of the sequencing itself. Number of total reads based on the rarest. Roughly what percentage of genes could we expect to detect protein genes of the rarest? What percentage of genes would we expect? Well, I mean, we're looking at being able to get not assemblies from every genome, but you'd be looking at getting a very, very good representation, maybe 5 times coverage. So you would hope that in terms of genes detected, you'd have a very high percentage of the total genes that are present. So So yeah, I get it from your metagenomic screening. So from the functional analysis point of view, you'd hope to get close to completely what's there. So I hope that answers your question, Paula. Okay. And Patricia in the Q&A, is there any way to rid off human genome from whole? Not quite sure I'm interpreting the question here. Is there a way to rid off human genome from whole metagenome sequencing? So yes, I think we kind of very briefly touched on that in one of the slides. You can certainly look for the amount of human contamination in the databases, and if you follow the protocols that come through, you should be able to minimize that depending on the quality of the sample taken and the way you do your extraction. So you can specifically extract for the prokaryotic genomes and not from the eukaryotic ones, and hopefully that helps clean up. But I can certainly provide more information to you, Patricia, if you want there, and provide any protocols that are available. Do contact me, and if you're interested, and let me know, and I'll be able to give you a much more complete answer to that.
Thanks, Paul. Can you hear me?
I can.
Oh, I've been trying to get on, but the internet is being a bit funny. So you've answered all the questions I can see in the chat and the Q&A, and these will all be collated together and put in the show notes on YouTube. So that will be available later on today. And when a reminder email comes out about the next webinar next week, I believe, to sign up for that, we'll also have the on-demand link in there for this week's webinar, as well as the one that went out again last week for the on-demand for week 1. So thank you very much, Paul. And please feel free to email through any questions that you might have after this, or comment on the YouTube video, and we'll get back to you with an answer and provide any additional slides or material that you might need. Okay.
Thanks, everyone. Maybe we'll see some of you in a couple of weeks' time for the whole genome plant animal sequencing guide. Until then,
yeah,
cheers.
Thanks for joining us, guys. Goodbye.
Bye.
Are we still here?